I am Dr. Joyita Giri, presently working as a DBT Wellcome Trust Fellow in National Institute for Research in Reproductive and Child Health, Mumbai. I did my doctoral study from the University of Calcutta Department of Biochemistry and postdoctoral research training from the University of Wisconsin-Madison Department of Medicine. My postdoctoral research aimed to identify the molecular mechanism of immunomodulation and tissue regeneration deployed by mesenchymal stem cells during catastrophic illnesses like colitis, cancer, etc. My laboratory was involved in the program for advanced cell therapy at UW-Madison, where my research outcome guided our clinical team to exploit MSC as a living cell drug in a human clinical trial. I have tackled two long-standing problems related to MSC-based therapy. One, how exogenously delivered MSC crosstalk with body immune cell to reduce gut inflammation, and two, how to optimize MSC's pharmaceutical effect in human clinical trial. My findings established the importance of viable metabolically fit MSC in a successful clinical trial. Now, culture-adapted MSCs are considered to be an excellent cellular pharmaceutical for various immune disorders. Nonetheless, the crosstalk between adaptively transferred exogenous MSC and host immune cells remain elusive. This inadequate knowledge hinders MSC-based therapy to reach the primary efficacy endpoint. In my study, I confirmed the beneficial effect of bone marrow-derived MSC in reduction of tissue inflammation in a mouse model of acute gut injury such as colitis. Using this translational model, I have mapped entirely novel observations. Host monocyte macrophage are essential for the MSC's effect to occur. Secondly, MSC secreted chemokine CCL2 and CXCL12 cooperate as a heterodimer to polarize macrophages to an IL-10 positive anti-inflammatory macrophage phenotype. And number three, polarized macrophages secondarily lead to IL-10 polarization of intestinal B and T cell, which further triggers reduced tissue inflammation. To our knowledge, this is the first report demonstrating the biological relevance of chemokine cooperativity in shaping the immune milieu of host tissue. Although preclinical studies strongly support the role of MSC as a living cell pharmaceutical product, only one such product has met regulatory approval by the European Medicine Agency. We hypothesize that this disparity arising out of interspecies outcomes may be related to cell drug deployment variables in human clinical trials. My extensive preclinical studies supported this notion and identified that MSC's viability, cryoinjury, and route of delivery are important variables in the inability of advanced clinical trials. I further discovered that only immune compatibility between MSC donor and recipient allows for clinically effective redosing. This study indicates an exciting possibility that allogenic MSC can be engineered before infusion to induce stable engraftment would be an effective way to enhance MSC's therapeutic potency. Presently, I am involved in deciphering the role of endometrial mesenchymal stem cells in patients with recurrent implantation failure, and we are hoping that this study in the long term definitely help to develop strategies for improvement of IVF success rate and designing novel contraceptives. This is a brief of my research work. If you want to learn more detail of it, feel free to contact me. Thank you.